So this raises a question. Why are people so interested in Archaea? It's not because there's a bunch of pathogens. That's the usual reason why people are interested in bacterial groups, right? Because there's a bunch of pathogens in them. There aren't any archaeal pathogens, so why? There are some important industrial processes that are driven by Archaea, and waste management is probably the big one, both the problems that it creates and the opportunities. But that's true of chloroflexes too, and not very many people study chloroflexes. So why? Why are people interested in it? Most work on Archaea is driven by this kind of limits of life thing, and the fact that Archaea are a great way to study organisms that we're more interested in, and by that, of course, I mean people. If you look at the phylogenetic tree with a root, and again, this root was generated by looking at deep duplications of genes, what you see is that Archaea are actually a kind of, they're a member of this eukaryote group. And they're both primitive and specifically related to eukaryotes. I hesitate to use the word missing link, but the way people normally think about a missing link is exactly what these organisms are. These organisms are not an evolutionary intermediary between these two, but they're a good surrogate for that. There are so many ways in which Archaea are similar to eukaryotes, but simple, and more similar to the ancestor than anything else that we have in our hands. But there's a lot to learn about where eukaryotes came from by studying Archaea. And so there's lots of interesting kinds of questions you can talk about. So where did the nucleolus come from? You guys know what a nucleolus is, right? It's the machine inside the nucleus where ribosomes are made. Bacteria have a nucleolus? Probably do, actually. They probably have one for each ribosomal operon. Because there's a bunch of stuff that has to happen though, right? You transcribe the RNA. Ribosomal RNAs are the most abundantly transcribed thing in any organism. And that RNA has to be processed, and the ribosomes have to be assembled. And if that's happening right there where the gene is, then that's a kind of functional nucleolus. But there are other things that happen in the nucleolus that aren't involved directly in ribosomal processing. tRNA processing occurs in the nucleolus. A bunch of RNA modifications have to take place. In fact, the nucleolus is kind of the RNA processing center of the cell. Archaea have a nucleolus for the very simple reason that they have these same RNAs, things like small nucleolar RNAs, spliceosomal RNAs, all that kind of stuff. And they're centered around wherever the ribosomal operon is. Most of these organisms have only a single ribosomal operon, so that defines the location of the nucleolus. I already mentioned RNA polymerase. RNA polymerase in these organisms works exactly like POL2 in eukaryotes, but you don't then have to worry about other kinds of transcription going on. So it's a great way to study transcription by POL2 in a system where you don't have POL1 and POL3 going on at the same time. Archaeal promoters have a title-box binding protein, but they don't have a lot of the other weird things that eukaryotes have. They don't have enhancers or anything like that, for example. The organisms are also, some of them, very primitive. So when you look at the bacterial world, there's some organisms that are a little bit primitive, things like aquifex, but nothing as primitive as some of the Archaea. So here's the last common ancestor. Here's aquifex. Here's Thermatoga. Here's Pyridictium. The eukaryotes, you're lost. I mean, these branches are all long. So if you're interested in looking at an organism that more than anything else alive today tells you about this last common ancestor, it's going to be organisms like Thermococcus and Paradicea. Those are the most primitive organisms we know about. Why are they primitive? Any idea? So that's an interesting idea. So the places these organisms live in are exactly the same as they were 3 billion years ago. Hydrothermal vents, right? Probably the reason, though, is that these really, really primitive organisms are also the most extremely thermophilic organisms we know about. And so think about how evolutionary change happens. Let's say you've got a protein and you've got 2 amino acids that interact in hydrophobic core of that protein. If you're going to change those amino acids, genetic drift, right? If they interact with each other, then when one changes, there's a selective pressure now to change the other one so that that the interaction is restored. There's a compensatory change in the other amino acids around it. This is really easy to think about for RNA structure where you've got a base pair. And if you're going to change that base pair, you change one base, and now you have a mismatch. And then either it reverts or the other base changes to restore the base pair. So you've got a transient mismatch or a transient defect in the molecule. If you grow at 30 degrees, no problem, right? But if you grow at 15 degrees, that's a problem. These molecules are not capable of withstanding these small transient defects. The result is the evolutionary rate of these organisms, particularly for RNA, but also proteins, is a lot slower. So these organisms, because of the environment they live in, are very slow evolving.